All right. Okay, I'm not going to start with my title slide. I'm going to go crazy and start with this slide. So here's a question for you early in the morning. I know we haven't had a lot of interactive talks, but we're going to make this one a little bit interactive. So can anybody give me a reason why so many colleges and grad schools teach that you should reject your hypotheses at a significance level of alpha 0.05? Any ideas of why we teach that?
Convention.
Because it's convention, right? Because that's what you do, right? Everybody here uses 0.05. Your journals use 0.05. Grants, use 0.05. So I posit a follow-up question. Why does everybody use 0.05? It is true that historically this comes from a Fisher paper where he said if you would see this less than 1 out of 20 times, then we should do further, investigation into the claim. But that's not the answer I'm looking for here. I'm trying to, get us thinking. The answer I'm looking for here, it's because it's what you were taught in college or grad school, right? And I am not going to spend my talk doing a philosophical analysis of Fisher or 0.05, but just kind of trying to get us thinking, like, we use this all the time, and whether it's a type 1 error rate, which is more of a probability of rejection, or a false discovery rate, which is more of a proportion of the number of false positives, this is a cutoff that is ubiquitous and really a part of all of the science that we're doing. Okay, so like I said, I'm going to be talking about false discovery rates. I'm going to be talking about online false discovery rates, which means sequential experiments. We've come up with this idea of sub-batching. A lot of this work is due to my former student, Lathan, who is at Merck and heading to Mount Sinai this fall for med school, actually. And the data that I'm going to be showing you comes from my biology collaborator, Dan Stobel, who's at Harvey Med College. I'll just also speak to the fact that I'm at a place called Pomona College. It's an undergraduate-only institution. It's actually in LA County, but like 55 miles from here, so it's like the farthest you can get in LA County. But it's a great place with smart undergraduates, and I get to do some really interesting research. But I love coming to CGSI because I don't have computational biology collaborators where I am. I'm actually in a department of mathematics and statistics. So, my collaborators are really on that end. So, love being here. Okay. So, as we have known from the last few weeks, we use p-values for all sorts of different things in computational genomics. But just to frame this talk, I'm going to be talking about differential expression analysis. So again, the data that I'm using is going to be RNA-Seq data. I'm really not going to talk about the data or the experiment very much because the work is, can be applied to any situation where you have lots of p-values coming in. So many of the sort of talks and ideas that were generated over the last few weeks. But just to, again, set the stage, give us some context, we're going to assume that we have some number, in the thousands, maybe 10,000 genes, and we've done an RNA-Seq experiment over 2 conditions, and we're trying to figure out which of the genes are truly differentially expressed and which of the genes are not differentially expressed. So, we want to find the ones that are actually differentially expressed and ignore the ones that are not changing over those 2 experimental conditions. And we're going to use p-values to control the false discovery rate. And a p-value is, for those of you who took statistics a very long time ago, the p-value is the probability of rejecting the hypothesis given that it's actually null. And the false discovery rate is the proportion of rejected hypotheses which are actually null. So the main difference between these 2 ideas is that the p-value is sort of conditional on the setting being the truth being null, right? So given that there's nothing interesting about this gene, what's the probability we'll find it significant? And the false discovery rate is sort of the opposite. It's the given that we found it significant, what's the probability that it's not what's the probability, but it's that, null idea. It's going to be a proportion, not a probability. But right, so we're conditioning. And for those of you who maybe haven't thought about conditionals a lot, and I don't know how many of you are basketball players in here, but if you think about the NBA, which is the Professional Basketball Association in the US, if you say, okay, given that you play in this Professional Basketball Association, what's the probability you're over 6 feet tall? And again, I know probably most of you don't use feet, but that's pretty tall. It's very high, okay? Given that you're 6 feet tall, given that you play in this Professional Basketball Association, the probability that you're 6 feet tall, the probability that you're tall is very, very high. Most of the basketball players are very tall. But if you switch that statement around and you say, given that I'm very tall, what's the probability I play professional basketball? It's extremely low. Okay, so these ideas of conditioning are going to, they're not actually super important to my talk, but they're important to the distinction between a p-value and a false discovery rate. One more kind of slide to get us thinking about what all these things are. We think of, M as this, number of 10,000 genes that we're considering over these experiments, and 4 things can happen, right? They can be truly differentially expressed genes. They can be truly not differentially expressed genes. And then when I go to do my experiment, I can either find them as significant, or I can say I didn't find significance. And the number we're really interested in this table is this letter V. This is like a total mathematician thing to do, right? I said the number we're interested in is that letter V, right? But those are the ones that really are not differentially expressed, but we found them to be significant in our experiment. And the 2 concepts I was just talking about above are, do I find any V's, right? Are there any of these false positives? Versus the false discovery rate, which is what is the proportion of false positives, right? And again, those are sort of 2 different concepts. And we're going to focus on the proportion of false positives. But in order to develop the tools that we need, we're going to start with that first thing. What's the probability of seeing a false positive at all? Okay, so many of you, I'm guessing, have seen this idea of a Bonferroni correction, but I'm also going to guess that most of you couldn't derive why it is that you divide your level of significance by your number of tests. So I'm going to tell you why. So we're going to start off with a situation where we only have 2 tests. So you have 2 genes in your experiment, and we're going to assume that both genes are actually null. So there's nothing exciting going on. You did a very bad experiment. It was bad on many levels. There were only 2 genes, and it was, null. So we can set the probabilities of, I think I can do this here. We can set these probabilities, oh, look at how fancy that is, of each of those genes being rejected, being called significant, to be alpha star. And what we'd like to do is bound the this mistake that we're talking about, this rejection mistake. We'd like to bound that mistake over the entire set of tests. So we'd like to say, okay, what's the probability that we're going to reject either one, right? Either the first gene or the second gene. And again, reaching way back into high school when you looked at Venn diagrams like this and probability statements, you might remember that the probability of the first thing happening or the second thing happening turns out to be the first thing plus the second thing, but then you've you've double counted that overlap, so you subtract it out. And you see that this number right here, this red question mark is unknown, but will always be non-negative because probabilities are always between 0 and 1. And so this Bonferroni approach here allows us to bound the probability that we get at least one rejection. At least one rejection right here will be less than or equal to alpha if we set the individual rejections to be alpha over 2. And that this argument easily scales to as many genes as you want. These Venn diagrams happily work for sorry, I have something in my throat, happily work for 10,000 genes. And so the Bonferroni control says for every single gene, just divide the alpha by m, your number of genes, 10,000, and reject any hypothesis such that this the p-value, that pj is the p-value, is less than the cutoff. And that will control the probability of making a type 1 error. Okay, and part of the reason that I wanted to tell you about this is because I wanted to motivate the idea of alpha spending. So alpha spending is this idea that we have this 0.05 magic, and we have to split it across many tests that we're doing. And what we just did in that Bonferroni is we said, okay, take that alpha, divide it by 10,000, and give 1/10,000th of the 0.05 to the first gene, 1/10,000th of the second 1/10,000th of the 0.05 to the second gene, etc. We've split that alpha. Bonferroni represents very conservative alpha spending because we didn't worry about that overlap. Turns out, there are lots of ways to consider that overlap that we're not going to do today. You may be familiar with alpha spending and other methods. So for example, clinical trials that look at the data halfway through, they do interim analyses. They do alpha spending with things called, O'Brien-Fleming or Pocock. Those are similar. Okay, but now I'm going to turn our focus to false discovery rates, which is really sort of what we've, been working on. So the data that come in, again to this context of RNA-Seq, and we've got, in this particular example, I was just simulating things. I just made this all up, but I have 500 null p-values, and I have 500 true p-values. And the true p-values are the tan ones, and you can see that those are right skewed, which means that they tend to be smaller p-values, which is what we want, right? Our true p-values, the ones that are actually differentially expressed, should be the ones that are small. And in this left-hand graph, this histogram of p-values on the left, it's two separate histograms. It's two separate histograms, a histogram of null p-values and a histogram of true p-values. And I've just plotted them in such a way that you can sort of see where their overlap is, but you should think of them as completely separately plotted there. Yes?
Where are there three colors?
Where's there what?
Three colors?
Oh, did I say three colors? I didn't mean to say three colors. Oh, why are there three colors? Sorry, there's three colors because that's where they overlap. It just these tan ones are sitting in front of the gray ones. That's it. So, it's terrible. So you have the tan histograms, and they're just literally, they're opaque, and they're sitting in front of the gray histogram, the histogram of gray null p-values, and you can see through the tan ones, and so you can see the gray ones in the back. Does that make sense? So there's really only two colors, it's just you can see them separately. And that's what we would love to see, and some of you may be familiar with the idea that when you have null p-values, they tend to be, or they theoretically are, uniformly distributed between 0 and 1, so that's sort of seen in this empirical simulation, that these gray ones kind of make this, sort of boxy shape instead of the right skew. But in real life, what we see is in the right. Right? We don't know when we do an actual study, we don't know which are the gray and which are the tan. What we see is just one uncolored, right? So what we would see here is that we would see a histogram like this one, and we wouldn't see these colors, right? We would just see a histogram of p-values where we got, a lot of them up, near this value, et cetera. And what we'd like to be able to do is break down and figure out which ones are gray and which ones are tan, okay, from the uncolored right-hand side. Okay, so now finally to this idea of false discovery rates. What is it, and how do we then use our information to control the false discovery rates? Just for notation, we're going to assume that the p-values are sorted. So p1 is my smallest p-value, and pm is my largest p-value. I have pi naught, which is my proportion of actually null tests. And so I can find the false discovery rate at any particular cutoff, right? So what is that going to be? It's going to be the number of p-values that are less than or equal to that particular cutoff out of the null tests, the gray ones, divided by the number of p-values that are less than that cutoff out of all the tests. That's what the false discovery rate is. Just, sort of by definition. It's the number of nulls divided by the total number of significance, or the number of false positives divided by the. We have that +1, so we don't end up dividing by 0, but the +1 is sort of irrelevant. So what am I saying? I'm saying that if we start off at some, C right here, like on this axis here, we're just going to count how many. I can't really get this to work. Okay, we're going to count how many are gray that are less than that cutoff, and how many total less than that cutoff. But of course, as I told you, I don't know how many are gray, and I don't know how many are tan. So we're going to approximate the numerator using this idea that the p-values are uniformly distributed. So if the p-values are uniformly distributed, then a really good estimate of the numerator of the false discovery rate is just whatever that cutoff is times the number of null tests. Okay, so if I have 500 genes and I'm wanting to know the cutoff of 0.1, well, about 50 of them, 10% of them will be less than the number 0.1. That's sort of what we mean when we say they're uniformly distributed. So I can approximate this numerator at a particular false discovery rate of pj. So that's saying, at a particular p-value, which is my cutoff, I can say that the false discovery rate approximation for it would be pj, because that's the cutoff, times the number of tests, which is m times the proportion. Unfortunately, I don't know that proportion. I don't know that p-naught. But I do know that it's a number between 0 and 1, so I can just say that the false discovery rate is less than the p-value itself times the number of tests divided by j. The denominator is j because if I'm rejecting all of my tests that are pj or smaller, that means I'm rejecting j tests because I've sorted my p-values, p1 through pj. So if I'm using the pjth one as my cutoff for rejection, that means I've rejected j things, which is why the denominator is j. There's a lot going on here. Anybody want to ask a question?
Could you repeat the equation?
Yes. Okay, so the numerator takes a cutoff value, and I'm sorry, the first equation is what I mean to say. The first equation tries to get in my head what is a false discovery rate. Well, it's the number of false discoveries divided by the total number of discoveries. So I calculate my false discovery rate at a particular cutoff. So in the top equation, I'm using a cutoff of just some arbitrary letter C. So I have to count how many things do I reject that are gray and less than C, divided by how many things total do I reject that are less than C. So now in this second equation, instead of using an arbitrary C, I say, okay, well, at a particular p-value, let's just say I'm using the 47th p-value that are ordered, sorted, right? The 47th p-value. Well, okay, if I use that one as my sort of criteria, then I'll say, okay, that means my denominator is 47. I know that I'm rejecting 47 of my genes. So what's my numerator? Well, my numerator is approximately whatever that p-value is times the number of null tests. The number of null tests is m times pi naught. That by definition, this is just notation that I'm using. M is the number of tests, pi naught is the proportion of null tests out of the total. But I don't know what pi naught is. It's part of the reason I'm doing all this. And so I know that pi naught is a number between 0 and 1, so I can just say that my false discovery rate is less than whatever that p-value is times m over j. Okay, I'm going to give you the equation again on the next slide. So there it is again. And that gives us an adjusted cutoff that adjusts then for every gene separately. So now, instead of having an alpha level, which is alpha over m, now my cutoff changes depending on where I am in my, series of tests. So again, I'm guessing that many of you have used this cutoff in your own analyses. You use a Benjamini-Hochberg cutoff where what do? You take your alpha level, you multiply it by j, and you divide by m. Or more likely, you use a function that says adjust.p method equals Benjamini-Hochberg, and you have no idea what it's doing, and it just does exactly this on the screen. But maybe one of the relevant points here is that for every gene, it's using a different criteria, right? And you'll notice that the very first gene uses the Bonferroni cutoff, right, alpha over m. And the very last gene uses just the alpha cutoff, uses the 0.05. I would love another question. Yes? Yeah?
Yeah, so like the first hypothesis has lower chances of being rejected than the second one, right? Even if, even if
The first gene has a lower bar to cross than the second one. Now, the first gene is a smaller number than the second one, or the first p-value is a smaller number than the second one by definition, right? P1, I've sorted my p-values, right? So P1 must be less than or equal to P2. But you're right that the bar that P1 has to cross is higher than the bar approximately twice, exactly. And there you go, Benjamini and Hochberg. Okay. Okay, so what I'm building to is an idea of controlling this false discovery for a series of experiments. Okay, so you might, the example that I'm going to do is going to talk about, I have these 6 bacteria experiments, and I want to control the false discovery rate over all of them. So I did a little thought experiment here. I said, what if I had 5 experiments, and they all had 10 genes each, and I controlled the false discovery rate, like I would in this, we call it offline way, but maybe this naive way where I controlled the false discovery for the first one using m equals 10, and then I control the false discovery rate using the second one, m equals 10, right, all the way through my 5 experiments, versus what happens if I did sort of the more accurate, hey, I'm actually doing 50 hypothesis tests here, and I should put them all together and control my false discovery rate using m equals 50. So here's this little thought experiment. So these are the alpha j values, the ones we just derived, j times alpha over m, in these 2 different settings. So in the black line are each of those individual cutoff. I've said the x-axis is the index of, quote, the sorted p-values, because in the correct, pooled experiment, they're sorted, p1 through p50. In the sort of incorrect, I sort the first 10 individually, then I sort the second 10, within that group, and I sort the third 10. And you can kind of see here that the mustard-colored dots have a steeper slope, right? Which means they're going to be less conservative. And I don't know if this image is going to work for you all, but what I've done is I've sorted the mustard-colored dots here. So that if you were to compare the mustard color to the black, it would be through these blue ones. Right, so this is, the blue dots are trying to get at this apples to apples comparison to notice that the black dots have a higher bar to cross. That the blue ones are less conservative. So if I do it in sort of this incorrect way, where I'm where I'm, looking to control the false discovery rate, in an offline way, one at a time, then I let myself sort of off the hook because I'm dividing by 10 instead of dividing by 50 is the gist there. So what is the problem? Well, the problem is that the theoretical control, which would be the black, is more conservative than the offline. And the other interesting thing here is that if I did these two, two different methods of analysis, I might actually get different genes being significant because I've sorted them differently, right? So I sort all 50 versus sorting in groups of 10, which kind of feels like a weird thing that makes me uncomfortable about the method, which is, sort of motivating what we're trying to do here. So we're trying to think about these sequential tests. How do we control the false discovery rate when we have multiple experiments coming in and possibly many experiments that we haven't even done yet? We don't even know how many experiments we might do in the future. So we have this second idea called alpha investing. We had alpha spending, which was getting rid of our alpha. And now we have alpha investing, which increases the alpha cutoff, every time a hypothesis is rejected. So the idea is that if you've rejected a lot of genes, well, then you don't feel like your type 1 error rate, your false positive rate, is particularly high. You have this sense that, oh, I've got a really powerful experiment, and I'm seeing a lot of significance across these two genes, or across these two conditions. So some of the original work on alpha investing was Foster and Stein, but I just wanted to point out this reference that the generalizing of the alpha investing to this idea of infinitely many tests was done by our CGS compatriot Saharon Rosset. So just and this gives us what we call online false discovery rate control. Okay, so I'm really going to be talking about these two middle bullet points. The first bullet point is the one I've already been describing. So the first one is where we control separately each of the experiments. So the second bullet point is with this idea of, okay, I get an experiment and I can control the false discovery rate, but I do it with the second batch in mind, second, third, fourth, infinitely many batches in mind. And sub-batch refers to an idea of I get an experiment and instead of looking at the entire experiment at once, I can break it up, maybe randomly into smaller pieces. So whereas a batch might be a physical experiment that you do, a sub-batch is a statistical thing where I randomly look at a few genes and I say, hey, did I get any rejections? Can I alpha invest at all? And then I look at my second sub-batch. So within a physical single experiment, I let myself spend and invest in smaller bite-sized pieces. Fully sequential is not something I'm going to talk about, but it's very intimately related to all this work. And it's where you look at a single gene at a time. So you do that alpha spending and alpha investing, a single gene at a time. This is sort of what I just said. Oops. Okay. So like I said, a batch is you can think of as an experiment. And we're going to set this alpha 1 for our first batch. And then depending on how many rejections we get, we're going to calculate that control value for the next batch. So we're going to do that alpha spending and alpha investing piece. And then we're going to continue. And it really doesn't matter how many batches we have in mind, because these methods allow us to iteratively keep going for however many batches. And even if the batch comes in the future, we can use the previous information. So how does this work? This is a lot of notation here. T is going to be the batch index And really this idea of alpha T is the key here, which is the significance level for the Tth batch. P is the p-values, N is the number of tests. R and R plus sort of measure the number of rejections. Okay, so this algorithm is due to Zrink et al. So this work is coming out of Michael Jordan's lab at Berkeley. And so what they've come up with are a handful of algorithms to do batch batch control. And okay, this is the algorithm. There's a lot going on here, but you can see that this idea of alpha 1, and then you iterate and you get your alpha 2, you get your alpha 3, and that this T, this batch number, could go on infinitely. So let me break it down a little bit for you. This first red that I've blocked out here, it's alpha spending. So the idea is that gamma, you can see from the sentence above that gamma is a number that's between 0 and 1. It says it's a non-negative sequence such that these infinitely many gammas would add to 1, so it's just going to be some kind of fraction. I'm really sorry about the cough. And so what I'm doing is I'm saying for this first test, maybe gamma is 1/2. So that would make alpha 1 at the 0.025 level, right? So we set it at 0.05. We set our alpha, our false discovery rate at 5%. And we say, okay, I'm willing to give half of that to my first test. And we see down below that we spend a little bit more. This is the iterative piece of spending, that this beta coefficient adds up how much of the alphas we've spent kind of along the way. So the beta coefficient that's being subtracted is saying, okay, in the previous batches, how much of that alpha have we spent? And so we've got these two ways of spending that alpha, but then we invest the alpha. So, we've got a piece here that says, depending on how many rejections we have, this is, the N is the sample size in the batch, and R is the number of rejections in the batch. And this number is going to be greater than 1, greater than or equal to 1. And so it's letting us invest that alpha, and we don't want to just spend the alpha, because if we only spend, then all of a sudden we're trying to reject at an alpha of basically 0, and so then all of our future experiments would be in the dust. So so what we see through this algorithm is that we come out with a control for a particular batch. Right? We come out with alpha 1, alpha 2, alpha 3, and I've got these, sub and superscripts such that, now we use the alpha level for the batch, which is alpha T, and we apply Benjamini-Hochberg. So we apply the, J times alpha T over M, and so then we reject any hypothesis in batch T where the p-value is less than or equal to this particular cutoff. And Zrink et al. have shown that for any batch that this particular algorithm, and they've got a couple of different algorithms, and they use Benjamini-Hochberg story and a bunch of different ones that they can control it at alpha. Yeah?
So how do you get the gamma sequences? What does that mean?
Yeah, so let me just... That's a great question, these gamma sequences. So you could do whatever you wanted, and the really aggressive. So it sort of depends on what you think your rejections are going to be in that test or in that experiment. What they recommend, and what we've done in our simulations, is kind of an aggressive gamma, where we let that gamma 1 equal 0.5 and then gamma 2 equal 0.5, and basically spend it all, but then have the investing kind of help the on the other end. So as long as you're seeing some rejections, then you can basically spend all of your alpha, or you could use any of your Calc 2 infinite sequences if you want to leave a little bit more and kind of have that be less aggressive, thinking that your later experiments might be the more powerful ones. But I think that's, this work is pretty new, and I think that's an open question, kind of in the sense of what types of work would be most suited for what types of gamma. Okay, and then this is their results. I don't want to spend a ton of time on them, but just kind of pointing out, one thing to point out, so both sets have power and false discovery rate. The left is the offline, so that original idea of just looking at false discovery rate for the first experiment, false discovery rate for the second experiment, et cetera. And then on the right is this batch Benjamini-Hochberg control. On the x-axis is the proportion of truly differentially expressed genes. Okay, so what we're seeing here is that in this offline control, in the left-hand side, that the false discovery rate kind of grows out of control. But if you're sitting in the front row, you can also see that the sample sizes here are quite small. So these are batches of size 10, batches of size 100, and then by the time you get batches of size 1,000, that actually you're controlling your false discovery rate in a totally reasonable way. So I don't know if that's a spoiler. One of the conclusions to my work is that when we're working with these huge data sets, that all of this fancy methodology actually doesn't change the results very much. But that's the last slide. Okay. So then Lathan came along and he took all the Zernick work and he wrote all of the batch algorithms into an R package, which is just a fantastic package. They've got really good references and whatnot. And he did some other analyses that are kind of interesting that I'm not going to spend a lot of time on. This is a very similar picture to the other one, but it's too small to see, and I'm not going to detail it. I'm just going to tell you what the main result is, which is that if you have some biological input, some knowledge about the experiments you're doing, and can order the actual experiments, order the batches in a meaningful way. You have some experiments that are very high powered, then the experiments themselves are high powered, then in conjunction with the batching false discovery rates, you can improve your power even more. So again, putting those experiments that are that have a lot of rejections in them, putting them first, does that in alpha investing and allows for really high powered experiments. So okay, so then I have this colleague, Dan Stobel, and this is just a small part of the experiment he's doing on. He's interested in sort of regulatory mechanisms like RPOS. And so this is a small piece of what he was doing. He took these 6 different bacteria species and made a really straightforward experiment. He just had them at 37 degrees in growth and then put them into 15 degrees Celsius, and after 3 hours measured them again. So really just this cold shock experiment. He had 4 samples in each of the 2 conditions and then these 6 different strains. So it was nice for me because it just really set up this idea of sequential tests. So we have 6 different experiments, and they all have differential expression p-values associated with them. So I thought I would show you what it looks like. These are in an arbitrary order. These 6 strains, they're in an arbitrary order, and you can see for this first one that I did that gamma of 1/2, which meant that alpha 1 was 0.025. And so, then I alpha invest, and this experiment actually has a really high number of differentially expressed genes. And so these rejections make these alpha values go up. And so we can see these cutoffs by experiment. But of course, when we go to reject individual genes, we have to then sort within a batch. and the black line now becomes the Benjamini-Hochberg cutoff value using alpha t for each separate experiment. So again, the black line is that alpha cutoff for each gene within an experiment, and the blue and mustard colored dots are the individual p-values. I've just cut the slide off so that we could see it a little bit better. They do keep going up, and they go all the way to the, like there are yellow dots here that go all the way to the end here, but the slide's unreadable if you let it go that way. Okay, so then I preempted. I told you, well, what happens if we use this physical experiment and break it up arbitrarily? The Zernk results, the proof of the theorem relies on the null p-values being independent of the other ones. Well, by randomly taking 10% of the E. coli, I'm not going to break any of that theory. So I can break the up the individual batches into these sub-batches and apply the exact same algorithm. And this is what we see. Again, the black line is the alpha cutoff, and then each of these blue and mustard colored dots is the p-values. And we actually do improve our power, but it's going to be very minor. So okay, we don't know any truth here, so we don't actually know what's going on. Are we getting more true differentially expressed genes, or are we getting more false positives? So we did a little bit of simulating. So what we're going to be simulating here, that very top line is the don't do any correction. So it's just alpha 0.05 on everything. So it's like literally, it's going to give me terrible false discoveries. So instead, let's zoom in a little bit on that smaller piece. On the crowded piece. So again, this is simulated data where I've set the proportion of differentially expressed genes. That's on the x-axis. On the y-axis, I'm measuring power. The top line here is that offline control. So that offline control is the very first one I told you, which is just, you look at your first experiment and control, and then you look at your second experiment and control. It's that naive way. The red line is that batching. And then these lines in between are the sub-batching. So we gain a little bit of power in those sub-batches. And I think the 10 sub-batches hidden under the 20 sub-batch or something like that. There are 4 lines in between there. You just can't see them all. There's 4 lines right here. It's just that one is hidden under one of the other ones. So it's not a huge gain in power, but it is modest. And then we ask the same question we ask about false discovery rates. Again, with our simulated data. And we see, wow, if we don't correct at all, if we just let 0.05, we're going to have a huge proportion of false discoveries. So let's zoom in on the crowded space again. And what we see here is that, again, that these small number of sub-batches, so having like 10 sub-batches, is a smaller false discovery rates than the offline method or the batching. Now, this is really striking because, in fact, it may be unbelievable that a method would have lowest false discovery rate and highest power, right? Usually those are kind of trade-offs there. So it's an effect that we're still working on. We're still trying to figure out. And indeed, if you put standard error bars on these graphs, you'll see that the crowded ones, they all overlap in terms of standard errors. So it's not clear whether the ordering of them is particularly significant. But it is true that the sub-batches do, the error bars on the red one do not overlap with the error bars on the sub-batching. So the sub-batching feels like it's getting much closer to, in both the power and the false discovery rate, feels like it's getting much closer to the offline work. Okay, and one last slide here to show the results on the biology experiment. So just looking at how many genes did we call significant across all 6 experiments. This is, for those of you who have not seen a plot like this before, this is similar to a Venn diagram. And what we did was we ran the analysis 5 different times. So the first run is the offline, which again is the naive one. The last one there is batch, where we're using that batch algorithm. We're just looking at one full experiment at a time with the Zernick algorithm. And then the 3 in the middle are those sub-batches, where we're trying to do that alpha investing inside of in smaller pieces. So for each of those 5 analyses, we say, which genes did you reject out of all 6 of these? We just have a big long list, which genes did you reject? And it turns out that they just reject the same genes, right? So like, almost, they all reject approximately 8,000 genes, and the overlap is the vast majority of them, right? So you can see that for example, like way over here, these 3 dots right here represent the genes that that the sub-batches rejected, that the other 2 methods didn't, and there's virtually none. Okay, so that's what it's saying here. The top histogram is measuring the number of genes that are in each one of these overlapping. It's like a 5-circle Venn diagram, and they're basically all rejecting exactly the same genes. So, and with that, thank you so much for having me, and I would be happy to take any questions.